Okay, this is the review video for Lecture 6, Biofilms and Microbial Genetics and Aspects of Genetics. We started with biofilms, and with biofilms, what's important for biofilms is their function. So what do they accomplish for the microbes? Where are they? Where do we find them? Where do we find biofilms? Make sure you look at that list of those places we find them. And the structure, always in biology, you're looking at a structure and how it leads to a function. You did that all through A and P, and you should still be doing that. How does the structure lead to a function? And so that's the things to know about biofilms. And generally speaking, we think of biofilms as forming on any solid surface in water. So like a river rock, we would expect to have a biofilm on it. In any place where there's a solid surface with a liquid, like teeth, the surface of teeth, or under rare cases, a blood vessel on a heart valve, something like that. But the bigger topic in this lecture was genetics. And we always start by reviewing the central dogma and remembering the relationship between DNA, RNA, and proteins. So make sure you've reviewed that. What is the connection between DNA, RNA, and proteins? What are they made of? What are their functions in a cell? And from that, we could look at different types of mutations. And in the slides, there's a slide that shows this, and I went through this in lecture, so it's on the audio and everything. But the main thing is there are 5 types of mutations, and you should know what they are and be able to name them if I describe them. Silent is a very common kind of mutation, and then missense is another. Nonsense is another. And then you can have insertions or deletions of any size, anywhere from 1 nucleotide to thousands or millions of nucleotides. And one of the reasons these insertions and deletions matter is some of them can cause a frame shift. And a frame shift mutation scrambles and randomizes a protein. Another question we could ask is, how do mutations happen? And the thing we thought about was a cell as it has its DNA and it copies its DNA. When it makes a new copy of the DNA, sometimes it makes a mutation. And so one of its daughter cells gets the original OK DNA. One of its daughter cells gets the DNA that has the mutation in it. And the daughter cells don't control which version they get. And the one with the mutation, that's all it has. It doesn't have the normal DNA, so it can't make the normal proteins. But another question is, do mutations happen in DNA or RNA? And in cells, they always happen in one of these. In a cell, if an error happens when making RNA, it doesn't matter. RNA is temporary and unstable, so it doesn't matter how bad an RNA is, it will disappear in an hour or something like that. But DNA is the only place where cells store information, and so if they make an error in copying DNA, that error is it. That is the new form of the DNA, and that is a mutation. So that's for cells. For viruses, for DNA viruses, it's the same, but for RNA viruses, they don't ever interact with DNA. Their genomes are RNA, and RNA is how they store their information. So if they make an error making their RNA, that is a mutation. So make sure you can understand that and walk through it. The next topic was error rates. And I did calculations with error rates, but you don't need to do these calculations. What you need to do is understand the units. So if I write 10 to the minus 5 errors per nucleotide, this is like saying 1 error per 10 to the 5th nucleotides. So that's 1 in 100,000. And so I want you to be able to compare a number like 10 to the minus 4 errors per nucleotide versus 10 to the minus 9 errors per nucleotide. Which of these leads to more errors and more mutations? That's something you need to be able to figure out. And another thing is, how common are mutants? So I won't ask you to do the calculation, but we calculated how many mutant virions would you find if someone coughed out during a COVID infection. And it's in the tens of thousands. Thousands of different random mutants get released every time someone with COVID coughs. And if you inhale a cloud of aerosols with COVID virions, you're not inhaling a bunch of identical virions. You're inhaling, well, lots of identical virions, but then also a large number of random different mutants. And that is a very important point when we think about infections. We're not infected by one bacterium and a bunch of its identical clones. We're infected by one bacterium, a bunch of its infected clones, and a bunch of random different mutants. And again, that is important when we think about evolution. Another thing we looked at was, why do we care about error rates? And if you look, examples I gave were for the smallpox virus. Its low mutation rate was one of the things that allowed us to eradicate it. Compare that to HIV, and its high mutation rate makes it impossible to even develop a vaccine. I get nervous every time I say this because many people are trying to develop vaccines for HIV, and they're always in clinical trials, but so far they've always failed. And someday I'm going to say it's impossible and someone will figure out how to do it, but I won't understand how they did it until I read how they did it, because they can't do it using the classical vaccine ideas, because its mutation rate is so high. Another thing is, can you compare error rates of eukaryotes, RNA viruses, DNA viruses, and prokaryotes? If you had to put these in order from fastest mutations to slowest mutations, or highest error rate to lowest error rate, you should be able to. There is a figure in the slides that lines them up correctly, so make sure you know that. And that it should make sense that eukaryotes in animals, we want the lowest mutation rate we can come up with, because we don't want our cells to be damaged. We want all of our cells to reflect our genetics perfectly. And chances are our mutation rate is just as low as we can possibly get it. Whereas something like a virus that's going to replicate super fast and any one virion doesn't matter very much, it may have an error rate 100,000 times higher than ours, or even 1,000,000 times higher than ours. Cool. Another topic is horizontal gene transfer. Horizontal gene transfer. And this is how bacteria swap huge amounts of DNA with each other. And there are 3 different mechanisms of horizontal gene transfer you need to know. One is transformation. One is transduction. And one is conjugation. So if I describe them, you need to be able to tell me the name. And then finally, what are the elements of natural selection? There are 3 conditions that must be present. And if they are present, natural selection will happen. The first one is heritable traits. So here, a trait is anything a protein might encode. So it might be an enzymatic ability, it might be the shape of a receptor, or whatever. Or in humans, it's these emergent properties that come from lots of different proteins. And there has to be genetic variation in a population, which is what I was just talking about. So a population could be that cloud of different SARS-CoV-2 mutants you sneeze out, or it could be the collection of different bacteria, all descended from one cell, but then there are a whole bunch of mutants during a bacterial infection. And then finally, a selection based on traits. And so these traits, in the case of a virus, we could imagine mutations in spike proteins change how well a virion might attach. And attaching is the essential step of viral replication. So if they have better ability to attach, they will replicate faster because they will attach to more cells in less time and ultimately be very competitive. So the heritable trait there is the spike protein. In a mutation, a new spike protein is encoded in the viral genome. That would be a heritable trait. And the selection based on the trait would be the fact that they can attach faster and therefore make more new virions of that type. And we also looked at antibiotic resistance in bacteria. The heritable trait would be the ability to survive the antibiotic. Genetic variation is everywhere in bacteria, and horizontal gene transfer is one of the biggest sources of that genetic variation. And then the selection based on the traits, antibiotics give the strongest possible selection because they stop bacteria from growing or they kill them. And a single mutant that can survive that has an infinite advantage over the ones that can't. And that's, again, the most powerful selection you can imagine is treating someone with antibiotics, and the bacteria in that person either are able to grow or they die. And we'll talk more about that in a later unit. So that was the review video for Lecture 6. I hope it helps.